All right, we are recording. Welcome, class. My name is Alberto Villarengo. You may refer to me as Alberto, Albert, or Dr. Villarengo. And welcome to food chemistry. If you have any questions or need to catch up on notes or something, we will have periodic breaks throughout the class that will be open for questions, answers, and catching up on notes. So let me do some screen sharing. All right. Can you see the presentation all right?
Yes.
All right. So welcome to my food chem teaching demo. Now, before I start, let me go here to this little blank slide and we're going to start with something. I want you to think of factors that affect food quality. And I want you to tell me about them or list them and describe them to me. What you believe is a factor of food quality.
Flavor, appearance,
Okay, let me rephrase. Food safety quality.
food safety would be probably pH, whatever microbiological testing is, so that it's been sterilized.
Okay, so pH and microbial content. Can you describe to me pH, please?
Uh, well, you need of a low. I think a low pH means more acidic, which has a less chance for bad germs to grow.
Very good. We'll get more into this stuff in detail later. I'm just wanting to get a feel for what you already know. All
Not too
right,
much.
what else? What else can you think of when it comes to like food safety factors or
Well, with microbial, I know that we used to do aerobic plate counts, and those were not a tell-all, but if it was low, then there was a lower chance of a pathogen in it.
okay? Very good. Any other factors you'd like to list out?
Those are the two that come off the top of my head right now.
Okay. Well, lucky you, you just listed a couple of intrinsic factors, which which we will be going over today already. So, hold on. Intrinsic food spoilage factors. That is the topic of today's lesson. Now, if you had listed some extrinsic ones, which we'll cover in the next topic, I would have listed them here, but on the right side of the slide. But it's okay; it doesn't matter. We're doing intrinsic today. Now, let me just see. Okay, there are many different intrinsic factors of food. Nutrient content. There's nutrient content. There's you already listed pH and microbial presence. Type. Nutrient content. There's the chemical redox potential. Okay. And then there's moisture and water activity. We will get into these bit by bit we'll get into each of these over time throughout the presentation. Any questions so far?
No, I remember water activity.
All right.
I do not know what redox is.
It's well, we'll get into that, but basically it's a shorthand term for reduction and oxidation, where molecules transfer electrons and cause reactions that way, chemical reactions that way. First intrinsic factor we'll cover is nutrient content. Now, there are 6 essential nutrients, at least to the human diet. Do you know what they are?
Calcium, I can guess, might be one.
Okay, that's a mineral. So minerals, you got one.
Okay.
Vitamins,
I wouldn't know that.
minerals, water, lipids, carbs, and proteins.
Okay. I don't know what a lipid is, but I'm sure you'll get to it.
Lipid is fat and oil.
Okay.
Now, nutrient content, the more nutrient there is on foods, the more spoilage bacteria are going to multiply. They get out, once they get on the food, they pick out on all the nutrients present in the food meant for us. And as such, they spoil the food, causing mold and decomposition, foul odors, and so on. Sometimes poison production, depending upon the type of bacteria present. Consequently, lower nutrition foods have a longer shelf life, consequently. And as such, higher. And as such, that's why like a lot of the shelf stable foods we see are like not like are like packaged and not considered healthy these days. What's the word? Junk food.
Okay.
Important note though, that doesn't necessarily mean they are low nutrient though. You see, a high nutrient content does not necessarily mean having all or multiple or a bunch of multiple nutrients. It refers to at least how much of at least one nutrient is present. I mean, all bacteria, if you just have a bottle of sweet water, bacteria will go crazy in that. Because you just got sugar and water. Those are like major nutrients and that'll attract bacteria like crazy. Now, not all bacteria are inherently bad. Some are actually good, and this leads to the microbial intrinsic factor you mentioned earlier. You see, some foods contain bacteria already as part of the process that they're produced. Not only can this bacteria is safe to eat, but it also competes with and keeps the spoilage bacteria from entering the food content. It's they can also be good for health, have pro health benefits when consumed as well. Important to note though that these friendly bacteria, if left unchecked, can they themselves become spoilage if they like multiply too much. It's all about monitoring and keeping things right. Some different foods that have these friendly bacteria that can have these friendly bacteria include kefir, which is like a yogurt-based drink. Yogurt, kombucha, a fermented cabbage vegetable product. Sourdough, but at least before it's baked gingerbread, and a few different types of cheeses, like blue cheese, for example. Blue cheese contains a type of fungus that keeps that although it makes the cheese super pungent and kind of little foul tasting depending upon your flavor palette, it keeps the bad bacteria out. You're retaining this so far?
Yes, yeah, I love blue cheese.
Okay. Okay, good. So that was a good example. Now, next is redox potential. You had asked about this earlier. Put into more detail, everything, us, food, our computers, everything, even food, is made of chemicals that transfer electrons and cause chemical reactions as a result of these electron transfers. When a molecule loses an electron, it's oxidized. When a molecule gains an electron, it's reduced. Some foods are, depending upon like what's their mineral content and such, and will have a tendency to transfer electrons, oxidize or reduce more readily. And as such, they will have a shorter shelf life because they're more prone to chemical reactions happening, causing spoilage. Such a thing even makes it easier for spoilage bacteria to actually break down these high redox potential foods more easily, making them spoil faster, not just easier. Okay, let's take a little breakdown. Any questions you got for me so far?
No, so losing electrons is oxidation, like when fish oil oxidizes and starts to stink.
Yeah, exactly. That's the kind of where that's the same kind of thing. Oxygen in the air actually called oxidation because oxidizing is known, oxygen is a known electron, is known to draw electrons to it. So when something is oxidized, it's usually the work of oxygen taking its electrons away. Fun fact, that's also what causes fish to stink, is when their oil oxidizes. Okay. Any notes you'd like to catch up on? Any note taking you catch up
I'm
on?
good so far.
Okay. Going to ask you a little brief couple quiz questions, make sure we're talking about. Name 2 types of foods that can contain competitive microbes.
I'm gonna take a guess here. Yogurt, I know, has living cultures in it. So,
Okay.
and I don't know what it is though. And same thing with kombucha, because I know that's marketed as a living drink. But I don't know if they're competing or they would just take out any bad stuff. I'm not sure.
Well, no, they compete with the bad stuff.
Okay.
If bad stuff tries to enter, they compete with it and force it out.
Okay. That's why in the Middle Ages, they used to drink beer because the water was contaminated. And the beer, I think it was made with yeast, would kill anything else.
The alcohol content the yeast produced helped too, but yeah, the yeast was big. Yeah. Okay. Another little quiz question. I want you to. Can you describe to me the difference between nutrient content and nutrient richness, or like nutrient
No,
wholesomeness?
I'm guessing that one of them means just a whole lot of it, and the other means several nutrients, not just one.
Very good. Okay. Yeah, okay. Nutrient wholesomeness or richness, which one was that? Would you say that is?
Richness probably means multiple nutrients, and content probably just means could be just one.
Very good. Okay. Last little quiz question before we move on. Why would bacteria let's see. Look, why would what are the 2 main nutrients that bacteria like to pig out on?
Oh, I wouldn't know.
You don't remember?
Probably sugars.
That's one of them, yeah. But sugar on its own can't be really digested because it's like doesn't have something to dissolve it in. It's very dehydrating. So the other,
Water.
very good. Okay. Make sure you're keeping good notes because this is all important stuff. In the lab, next classes, in the next session, we're going to go into the lab and we're going to not just analyze these different intrinsic factors, but we're going to tinker around with them to influence them and make food products shelf stable. All right, so make sure you're taking good notes, class. All right, moving on. pH and acidity. Earlier you had mentioned pH has to do with like acid content and such. Well, here's a little you're not far off, but there's a little difference there. You see, pH is all about the releasable hydrogen ions from a molecule, which defines a molecule as an acid. However, not all hydrogens on a molecule can be released as such, even on some acids. Basically, and as a result, pH does not necessarily measure full-on acid content. It basically measures all the hydrogen ions present that are loose in a solution. All right?
Okay.
You got okay. Basically, you can calculate this in a basic level as basically take the concentration in moles per liter of your hydrogen ions, your H plus ions, and then you take the negative log of them, negative log 10 of them, and that gives you your pH, at least basically. Yeah.
Well, I'm sorry, I got a question. Can you give me an example of a negative log base? I know what log base 10 is, but can you give me an example of a negative log base 10 of something? Because you lost me there.
Okay, well, let's say you have let me pull out my calculator. You know, let me pull out the calculator app. Hold on a sec. see. All right, can you see the calculator okay?
Yes, I can.
Okay, let's
It's
see.
great.
Long 10. Okay. Let's go with a concentration of 50 moles per liter. Just for example. Then take the log of that. That gives you a that would be a pH of 1.69 and so on.
Okay.
Then, or hold on. Hold on, that. Okay. Let's go 50. That's like the positive log now. Take the negative and go looking. I might have to brush up on this. This is a poor example of my demonstration. I'm sorry.
That's all right. We can repeat this if you need to.
All right. Well, either way, this goes to show the importance of a teacher knowing their stuff as well as being able, in order to be able to teach. Anyway, either way, pH. So basically, you're taking the concentration and you're taking the negative log of it. Wait, hold on. I think I figured this
So
out.
then so you would have a number less than 1, and then that would produce a negative logarithm, and then you negative it to make it positive. So try 0.4 moles per liter.
0.4 moles per liter. Okay.
Yeah, try that.
Negate. Long. Oh wait, wrong
No.
way.
Go 0.4, hit log, and then make it negative, which would make it positive.
Oh yeah.
So that'd be 0.3. A
Yeah,
pH of
good point.
0.3 would be pretty nasty, but yeah.
Okay, yeah, that's it. You see, most molarities actually will not be that high as like 50 or whatever. Most molarities will be like your in decimals as like not whole numbers.
Right. Yeah. So a pH of 5 would be anti-log that would be something like 0.00001 molar moles per whatever.
Yeah, exactly. And that shows you how little hydrogen, that goes to show you how small the
Yeah,
hydrogen concentration is, making that less acidic.
that makes sense.
Now it's important to note that pH itself is it's a unitless number. It doesn't
Right,
have any
because it's
units with
percent
it.
hydrogen. So a percent is unitless. Yeah.
Now, although pH is not necessarily the same as acidity, it does also dictate whether or not a food is considered high or low acid.
Okay.
Food that is a high acid food are usually very shelf stable, and they will have a pH equivalent to or less than 4.6. Whereas a food with a higher pH of pH higher than 4.6 is lower acid, and they are not as shelf stable. Such examples of high acid food would be like your citrus fruits, whereas your low acid foods or like your low acid foods would be like your milk or something. Another interesting thing is sometimes when during processing, acid can be added to food. And as such, it'll not only make the pH lower, but it also increase the water activity, but that won't matter so much because the pH is too low for any spoilage microbes to multiply.
Okay.
Now that pays into like tinkering around with the different intrinsic factors, which will, like I said, we'll do that in lab at next session. Now we went over pH, but acidity itself involves not the concentration of the hydrogen ions, but the actual acid that releases the hydrogen ions. Like, for example, sulfuric acid, which obviously is not added to food, but let's use that as an example. Sulfuric acid contains 2 hydrogen ions that can be released. So for every So basically that would mean that if you had pure sulfuric acid, the acidity would be in a solution. The acidity would basically roughly be half the hydrogen concentration.
Okay.
Assuming the sulfuric acid releases both its hydrogens at the same equally readily, which it doesn't, but this is a hypothetical example. So acidity is not the same as pH, and as such, it's based on the concentration of the acid that releases the hydrogen ions. Now this one, although like pH, the initial calculation is based off the moles per liter. But unlike this, with pH, this one stays at moles per liter and is referred to as molarity. Sometimes, let me make a little thing here. You see that M I just added?
Yeah.
That means molarity and is basically a shorthand way of writing moles per liter.
Oh, okay.
Now, if you want to influence, like I said earlier, if you want to influence the pH and acidity, you can make the food acidified. Add more acid to it, making it a higher acid food, increasing the pH and the water activity. And
Like
that
lemon
brings
juice.
us
When
to our
people add
next
lemon
slide.
juice. As
What?
an example, when people add lemon juice to stuff.
Yeah, exactly. There you
Or
go.
vinegar. Or vinegar also.
Yeah. Like you ever have that little science experiment when you were a kid and you're like adding lemon juice on an apple to keep it from going bad?
Yep. And you put that on the dried apricots to prevent them from getting dark.
You know your stuff. There you go.
No, those are old examples of when we used to dry apricots at home because I could not take
Oh,
sulfur. Sorry, I'm getting off track. Please continue.
either way, you'll have to tell me more after class. All right. As we were going off on how adding acid solution increases the water activity. Well, water activity, some people confuse water activity and moisture content, so I'm going to clarify that real quick. Moisture content is written as a percentage. It's the total mass of water that is a part of the total mass of the food altogether. So you can have like, let's say you have 100 grams of cheese. And then out of, after you dry it out, you find that the mass of the cheese, after removing all the water from it, is now, let's say, 80 grams. So do the math, you get 20 grams of water as part of that initial 100 grams of cheese. Your moisture content is 20%. And you getting this so far?
Yes, yeah, I remember this from my days working in the food industry because we had percent moisture, which is what you have there, and then water activity, and they were very highly correlated. But each food product was different because some foods had the ability to retain more water and have less free water.
Yeah, there you go. Because yeah, not all of these correlated, they can confuse, and that's good that you know this stuff. Makes it easier on me. You see, moisture content can include free water and water that's bound in the food. But water activity is just the free water, the water that's in the food, but isn't bound to it. It's the water that can freely flow in and out of the food. As such, water activity is measured with vapor pressure. And see how it's written as AW? That's like the shorthand notation for water activity. Basically, you take your vapor pressure of the water coming off the food, and then your vapor pressure of pure water, and then you divide the vapor pressure coming off the food by the vapor pressure coming off pure water, and that gets you your water activity. Some will leave it as a ratio like that, others will divide it out into its corresponding decimal form. Either way, it's important to note water activity and moisture content are different things, but they still both affect food spoilage because spoilage bacteria also need water in order to survive and multiply and such. Without it, they just shrivel up and dry up, or they turn into, or if they're a spore former, they'll just turn into a spore. All right. So moisture content versus water activity, similar. both essential. All right, we're going to close down now. Here is your last chance before we leave, before I dismiss you, to ask any questions, catch up on any notes, or get any clarification. So ask away if you have any.
Not now, but in the future, I still need a mathematical presentation to learn what exactly a mole is, how it relates mathematically. But that is future discussion.
Okay. Yeah, we can cover that in a little bit. Come see me. Well, come see me in the hotel room afterwards. And that's talking real life, not the demonstration. But okay. Back to the demonstration talking. All right, so you've got your okay. So you want to learn about mole math conversions? We'll get to that. In the meanwhile, next class we will have a laboratory. We will be doing a lab in which we will be analyzing, quantifying, and such and influencing the intrinsic factors we covered. This will allow us to not only determine how likely a food is to spoil, but also allow us to influence said food to make it more shelf stable. Please remember to bring a notebook and pen, safety glasses and shoes, a lab coat, and a scientific calculator. Everything else will be provided by the lab.
Cool.
That, unless you have any more last-minute questions or statements, class is dismissed. You may sign off.
Thank you.
All right. I'm going to keep recording. You can actually sign off now.
I'm working on it.
Okay. That was my demonstration of how I would conduct a class, save for a few little errors, like when it came to the calculating the pH. I obviously would need to brush up on that if I was teaching.
Sure.
Um,
And also you say come in my office hours, because right now we're traveling and we have we're at a motel, but yeah, come by my office hours.
well, no, I wasn't saying that as part of the demonstration, but
Well, I know. Okay. Do I leave the meeting?
yes, that's what I want you to do. All right, so like as I was saying, was my office hour. That's my demonstration on how I teach a class with a little bit of input from real life. I'd like to thank my dad who participated as my student. And yeah, that's my demo for how I'm teaching.